Good morning, my name is Ingrid Campo and I am going to talk about dengue disease. First, we need to know about the definition of the disease. Dengue is an infectious viral disease transmitted by the bite of infected female mosquitoes, primarily Aedes aegypti. Signs and symptoms of dengue. It is characterized by high fever, severe headache, joint and muscle pain, and rash. It is not transmitted from person to person and is common in tropical and subtropical areas. Signs and symptoms of dengue. There is some warning signs. We need to seek immediately medical attention. These signs generally appear when the fever subsides between the third and seventh day. Severe and continuous abdominal pain, persistent vomiting or vomiting blood, bleeding from the gums or nose, or blood in urine, dizziness, fighting or changes in level of alertness, lethargy, irritability, difficulty breathing. Causative agent. The family Flaviviridae is a large group of viral pathogens responsible for causing severe disease and mortality in humans and animals. The family consists of three genera: Flavivirus, Pestivirus, and Epacvirus. The Flavivirus, Latin flavus, meaning yellow because of the jaundice induced by yellow fever virus, comprise a large genus of medically important arthropod transmitted enveloped virus with more than 70 members that include dengue virus. Symptoms of flavivirus infection can range from mild fever and malaise to fatal encephalitis and hemorrhagic fever. Causative agent. Dengue virus is a single-strand positive sense RNA genome consisting of 11 kb in length. The single predicate polyprotein is translated by the genome that it is used by the virus encode protein into 7 non-structural proteins and 3 structural proteins, which are defined as follows: non-structural gene 1, NS1, non-structural gene 2A, NS2A, NS2B, NS3, NS4A, NS4B, NS5, and the structural protein including capsid protein, envelope protein, and membrane protein. Some of the non-coding regions are also present on the 3' end of the genome. Virus attributes. Structure and gene. It is a positive sense single-strand RNA virus with a spherical envelope. Serotypes. There are 4 distinct jet-related serotypes: DEMV1, DEMV2, DEMV3, and DEMV4. Immunity. Infection with one serotype confers permanent immunity against that specific serotype, but only temporary and partial protection against the others. Reinfection and severity. And second infection with a different serotype increases the risk of developing severe dengue. Pathogenicity. The virus primarily infects microvilles and monocytes replicating within white blood cells. The structural proteins plays a dynamic role in the structure of viral proteins, and the non-structural protein plays a vital role in virus entry, replication, assembly, and pathogenesis in the host, where they cause disease and pathogenesis. Mechanisms of infection. Regarding DENV infection and replication, there are numerous mammalian cell receptors reported to be mediate DENV attachment and entries such as DC sign, L sign, heparan sulfate, mannose receptor, laminin receptor, dopamine receptor, and others. In the early phase of DENV replication, virus particles will attack to the receptors and further enter the cell via the clarithin-mediated endocytosis pathway. The virus merges with the endosomal membrane once it has reached the interior of the cell and is then released into the cytoplasm. When the virus particles split apart, the viral genome is released. The viral genome is replicated after the translation of the viral RNA vRNA into a single polypeptide, which is then divided into 10 proteins. When structural proteins and freshly synthesized RNA protrude from the endoplasmic reticulum, virus assembly takes place on its surface. The trans-Golgi network is the place where the immature viral particles mature and change into their infection form. At that point, the full developed viruses are expelled from the cell and are free to infect additional cells. Pathogenesis and virulence factors. Transmission and dissemination. The virus enters through a mosquito bite and initially replicates within dendritic cells and macrophages, subsequently disseminate through the lymphatic and circulatory systems. Antibody-dependent enhancements. In secondary infections involving a different serotype, pre-existing antibodies fail to neutralize the virus. Instead, they form complexes that facilitate its entry into monocytes, thereby exponentially increasing viremia and the inflammatory response. Cytokine storm. Massive infection triggers an excessive release of inflammatory cytokines. Tumor necrosis factor, IL-6, and others, leading to increased vascular permeability, plasma extravasation, and shock. Endothelial dysfunction. The virus infects or disrupts the endothelium, resulting in thrombocytopenia, low platelet count, and hemorrhage due to cellular apoptosis and vascular barrier dysfunction. Dengue virulence factors. NS1 protein, not structural, a K factor released into the bloodstream. It causes dysfunction of the endothelial glycocalyx, increasing vascular permeability and plasma leakage. Glycoprotein E envelope mediates the binding of the virus to host cells and membrane fusion, which is essential for infectivity. Protein M induces apoptosis in immune cells, thereby facilitating viral propagation. Evasion of immune response. The virus inhibits the host interferon production, delaying antiviral defense. Genomic variation. Certain strains are associated with higher virulence and hemorrhagic manifestations. Epidemiology. Dengue virus causes the disease and burdens in most tropical and subtropical regions of the world, mainly in the Caribbean, Central and Southwest Asia, and South America. More than 100 countries are affected by dengue virus all over the world each year, and there is a high risk of infection with approximately 3.6 million people live in these countries. Over the past 20 years, the number of dengue cases reported by 505,430 cases in 2000 to over 2.4 million in 2010 and 5.2 million in 2019. The number of reported deaths increased from 960 in 2000, 4,032 in 2050, mostly affected younger age groups. In 2020 and 2021, both the overall numbers of the cases and the number of reported deaths appear to be declining. The data is still incomplete, and the COVID-19 pandemic may have made it more difficult to report cases in some nations. Global incidence and prevalence. Annual cases. It is estimated that there are between 50 and 400 million dengue infections per year. Population in BRICS, approximately 4 billion people, nearly half of the world population live in endemic areas, primarily in tropical and subtropical regions of Asia, the Americas, Africa, and the Pacific. Trend. Incidence has increased significantly over the past 3 decades, accompanied by an increased disease burden that approximately doubled between 1990 and 2021. High risk of severe dengue. Secondary infection, reinfection. Individuals who are infected a second time with a serotype different from that of their initial infection face a significantly higher risk of developing severe symptoms. Children and infants, children under 15 years of age, particularly infants, are more prone to developing severe dengue due to the increased permeability of their microcirculation. Older adults, individuals over the age of 65 are higher risk of complications. Transmission mechanism and vector. The Aedes aegypti mosquito is the primary vector of transmission in tropical and subtropical regions. Secondary vector. Aedes albopictus can also transmit the virus. Transmission cycle. The mosquito becomes infected by biting a viremic person, and following an incubation period of a few days within the insect transmit the virus to healthy individuals through the saliva. Aedes aegypti live inside homes and their immediately surroundings, preferring accumulate clean water in which to lay its eggs. Incubation period, generally 4 to 10 days following the mosquito bites. Culture and diagnosis. The diagnosis of dengue is based on virological tests, RT-PCR, and S1 antigen during the first 5-7 days of fever, and serological tests After the 7-day viral culture using mosquito C36 or mammalian virus cells, confirm infection by isolating the virus, although it is time-consuming and is used primarily in research settings. In summary, if the infection is very recent, 1-3 days, the NS1 test is the most appropriate, whereas after the first week, IgM is sought. Methods. Cell culture techniques are employed, often utilizing high-density bioreactors to maximize production. These methods are rarely used for clinical diagnosis. Antigen tests or PCR are more commonly used instead viral replication. The virus lacks independent life and requires infecting a host cell in order to replicate, utilizing a key protein known as NS3 for this process. The cultivation of dengue virus is a complex process used in research to study viral replication, evaluate antivirals, or produce vaccines, typically employing mammalian cells, such as virus cells or mosquito cells lines. The virus replicates within a host cell, releasing its RNA genome to create copies. Prevention and treatment. The mosquitoes that transmit dengue are activated during the day. To reduce the risk of contracting dengue, protect yourself from mosquito bites by using clothing that covers as much of the body as possible. Mosquito nets ideally treated with insect repellent if sleeping during the day. Window screen, mosquito repellents, and mosquito coils and vaporizers to prevent mosquitoes from breeding. Implement environment management and modification practice to prevent mosquitoes from accessing air-lying habitats. Properly dispose of solid waste and remove artificial habitats that may accumulate water. Cover, empty, and clean household water storage containers weekly, and apply appropriate insecticides to outdoor water storage container. If you contract dengue, it is important to rest. Manage dengue at home. Mild symptoms. Hydration. Drink plenty of water, oral rehydration solution to replenish fluids, medication. Take a paracetamol for fever and pain following the doses recommended by a doctor. Rest. Observe absolute rest to allow the body recover. Mosquito controls. Use repellent on mosquito nets during the febrile phase to avoid transmitting the virus to others. Avoid aspirin and ibuprofen as they increase the risk of bleeding. Treatment of severe dengue. Severe dengue requires immediate hospitalization. Hospital treatment includes intravenous fluid and electrolyte replacement, continuous monitoring of blood pressure and vital signs. In severe cases, a blood transfusion may be necessary. Early and appropriate treatment drastically reduces the risk of mortality. This is the reference of my work. Thank you so much.